Hi, I'm Sabrina Benitez.
I'm Sofia Escobar.
And I'm Juan Pablo Flanagan.
We're students from McAllen, Texas, and we're part of the International Baccalaureate program at Lamar Academy. And we are studying the effects of the perchlorate ion in simulated Martian soil on Solanum, lycopersicum, seed germination, and microgravity.
Hello, everyone. I am happy to be here. And now I will discuss some of the inspiration and the main ideas that came about in this experiment, particularly with regard to how we formulated this research question. So first, I would like to point out the recent discovery of hydrated salts on Mars. These hydrated salts, or perchlorates, particularly magnesium chlorate, magnesium perchlorate, and sodium perchlorate, were found in what is described as downhill water flows, otherwise known as recurring slope lineae, on the surface of planet Mars. These discoveries were made using A, the Mars Reconnaissance Orbiter, and B, high-resolution imaging. Interestingly, this detection was first pointed out by an undergraduate student at the University of Arizona in 2010. Second, the 2015 film The Martian, directed by Ridley Scott, and its realistic depiction of space also inspired us. This movie tells the tale of astronaut Mark Watney and his survival on planet Mars utilizing botany to create a successful and ingenious potato crop. So his effective use of botany inspired us to select the tomato seed. And although we chose a crop, we chose the tomato because of its fairly quick germination process, which is about 5 to 10 days. And lastly, the image of the Hawaiian volcano, Mauna Loa, represents the source of the Martian soil simulant, which we are using for our experiment. Specifically, the soil we are using came from the Pu'unene cinder cone portion of the volcano in Hawaii. And this soil is the closest soil that can be found on Earth to the Martian soil that was acquired in 1976 with Viking Lander 1. So there you are. Those are some of the factors or ideas that led us to formulate our research question, which is, how does the perchlorate ion in simulated Martian soil affect Solanum, lycopersicum, seed germination, and microgravity?
So our experimental design utilized the Type 3 FME mini lab. So in Volume 1, we had the soil with the 15 tomato seeds. Volume 2 contained distilled water, and Volume 3 contained the fixative formalin 10%, so that we can impede the growth of the seeds so that we can analyze them when they come back to Earth. So our experimental design consisted of 3 different groups that simulated different conditions because we had 2 independent variables, the soil type and the gravity. So Group 1 was our control group. So Group 1 had seeds growing in Earth gravity and Earth gravity and Earth soil. And Group 2 would test seed germination in Earth's gravity, but we would use Martian simulated soil with sodium perchlorate. And Group 3 would test seed germination in Martian simulated soil and in microgravity, and that group would be sent up into the International Space Station, and that would isolate the variable of gravity, and Group 2 would isolate the variable of the soil type.
So after the completion of the experimental groups comes the most important part of the investigation process, which is analyzing the results. Now, because of the limited shape and volume of the FME Minilab, and because of the discoloration effect of formalin, which is the fixative that we are using, there's not a lot of physical characteristics that we can analyze that will tell us much about the effects of microgravity and perchlorate on the seedlings. But one of the first things that we're going to do, it's very simple, and we're just going to count how many seeds germinated in space and compare them to how many seeds germinated on Earth with Martian soil compared to Earth soil. And then we will dry and weigh each of the seedlings and compare their weights, which will give us a very rough estimate of how much water and nutrients were absorbed by each of the seedlings. And then we will measure the radical length. The radical is what will become the root in a fully grown plant. And this will give us also a good idea of how the maturity and the growth of each of the seedlings was affected by microgravity and perchlorate.
So we hypothesized that Groups 2 and 3 will produce shorter radical lengths and limited seed growth because of the high solute concentration in the soil. And we also hypothesized that due to microgravity, group 3's lateral root growth would be evenly distributed along the radicle rather than concentrated at the end like most seeds grow here on Earth.
So now we want to talk about some of the challenges that we had during the ground truth trials. And one of them is finding a proper soil type. The first soil type that we used was common soil that we purchased at a local nursery, and it turned out to be very chunky and dry. And when we put it into the mini lab, it acted like a stopper, so the water couldn't properly flow through the soil and into the seeds. And then we switched to a much finer soil that we found, which was to grow cilantro in the kitchen. And then we sifted it to make it even finer and more suitable for the mini lab type. Another challenge that we faced was the seed count. At first, we only had 3 seeds, but we figured because of the low percentage of growth of tomato plants that we were probably going to need more. So we conducted several experiments to optimize the seed count and ended up with 15. And then we also had the trouble with determining how much water we would need. Too little water wouldn't catalyze the germination of the seeds, but too much water could also drown them. So we also conducted several trials to optimize that volume. And finally, just the FME mini lab size itself, the limited volume, which wouldn't allow for a lot of seeds or too much soil, that would limit the amount of water that we could use. The size also limited the extraction of the seedlings without destroying them, and it limited the dispersion of the water through the soil.
And finally, we'd like to give a special thanks to these sponsors. Thank you.
Any questions for this team?
Hello, my name is Mia. I'm from iLead in Southern California. I'd like to know how you determined that the material you obtained from the volcano was similar to Martian soil.
Recently, there was an experiment conducted by NASA on the same volcano, the Mauna Loa volcano, to test Martian conditions and living on Mars. And there on that experiment, they grew tomatoes, so that's why we chose to grow tomatoes. So when we bought the soil from that volcano, we decided to use that volcano because we knew that researchers from NASA were already using that soil for similar.
What sterilization procedure did you use to sterilize your seeds?
I'm sorry?
How did you sterilize your seeds?
Oh, well, we haven't conducted the experiment yet, but we're going to do a standard drying in an oven.
Thank you.
Thank you.